Welcome back 12th class. This is your chapter 12 biotechnology and its application. In our previous section, we have studied about biotechnological applications in agriculture. Now I am taking biotechnology applications in medicines. So let's start. The recombinant DNA technology is used for production of therapeutic drugs which are safe and effective. It avoids unwanted immunological responses commonly observed with similar products isolated from non-human source. About 30 recombinant therapeutics have been approved for human use in the world. In India, 12 are being marketed presently. Human insulin production by recombinant DNA technology. We have already prepared genetically engineered insulin. Insulin contains two short peptide chains, chain A and chain B, linked by disulfide bridges. You can see here the bridges. In mammals, insulin is synthesized as a prohormone that needs to be processed to become mature and functional hormone. It contains an extra stretch called C peptide. You can see here. This is extra stretch or free C peptide. C peptide is absent in mature insulin and is removed during the maturation into insulin. Earlier, insulin was extracted from pancreas of slegated cattle and pigs, but some patients begin developing allergies. So there is some disadvantages also. Disadvantages of extracting insulin from animals was insulin being a hormone is produced in very little amounts in the body. Hence, a large number of animals need to be sacrificed for obtaining small quantities of insulin. This makes the cost of insulin very high, demand being manifold higher than the supply. Slaughtering of animal is not ethical. There is a potential of immune response in human against the administered insulin which is derived from animals. There is possibility of sluggard animals being infested with some pathogen which may contaminate insulin. So, after that, production of insulin by recombinant DNA technology was achieved by American company Eli Lilly. This is Eli Lilly. In 1983, it prepared two DNA sequences corresponding to A and B chains. You can see in the diagram here. This is A chain and the another chain, another plasmid have the B chain. So A and B chains of human insulin and introduce them in plasmid of E. coli. Here you can see in this diagram, A chain introduced in one E. coli, plasmid of one E. coli, and the another B chain introduced into the plasmid of another E. coli. The A and B chains produced were separated, extracted, and combined. These were separated and extracted and combined by creating disulfide bonds to form human insulin called humulin. There is disulfide bonds. So after the production of this, it was very easy to produce the insulin. Next slide shows that how the insulin purify. There is a bacterium and this is the plasmid, and gene of interest introduced into the plasmid, and after that cloning, for the cloning process there is introduced into the bacteria, and after that there is a insulin protein production of insulin production, and after that there is a fermentation process, purify of insulin, and there is insulin ready for the distribution. Next slide shows gene therapy. Gene therapy is a collection of methods that allows correction of gene defects diagnosed in a child or embryo. By insertion of normal genes, the defective mutant allele of genes are replaced and non-functional gene is compensated. For the first time in 1990, a gene therapy on a 4-year-old girl with adenosine deaminase ADA deficiency. ADA is caused due to the deletion of gene for adenosine deaminase. In some cases, it can be cured by bone marrow transplantation and enzyme replacement therapy, but it is not fully curative. In gene therapy, lymphocytes from patients' blood were grown in a culture and functional ADA cDNA was introduced in these lymphocytes using a retroviral vector. For permanent cure, gene isolated from the bone marrow cells producing ADA at early embryonic stage can be possible cure. Other diseases like cystic fibrosis, hemophilia, cancer are also treated by gene therapy. So you can understand by this diagram, bacterium carrying plasmid with cloned normal human ADA gene, genetic disabled retrovirus, cloned ADA gene is incorporated into virus. Then the retrovirus infect T cells transfer ADA gene to cell. Then these cells are grown in culture to ensure ADA gene is active, and this active ADA gene introduced into the embryo or child. Genetically altered cells are reimplanted, produces ADA. Next slide shows molecular diagnosis. Early detection of a disease is not possible by conventional diagnosis method. Some technique used for early diagnosis are PCR, polymerase chain reaction. In polymerase chain reaction, low concentration of pathogen in the body does not allow in detection. The nucleic acid of the pathogen is amplified by PCR for its detection. It is being used for the detection of HIV, a suspected AIDS patient, and genetic mutation in the suspected cancer patient. Second techniques we can use in molecular diagnosis, recombinant DNA technology. A single strand DNA or RNA tagged with a radioactive molecule is called probe. In this method, a probe is allowed to hybridize to its complementary DNA in the clone of cells. The cells are then detected by autoradiography. The cell with muted gene will not be observed on the photographic film because the probe was not complementary to the muted gene. And the next technique used in molecular diagnosis is enzyme linked immunosorbent assay, ELISA. It is based on antigen and antigen antibody interaction. Either the presence of antigen are detected or antibodies produced against the pathogen are detected. So this was the molecular diagnosis. In another section of our chapter, we will take the transgenic animals. Thank you so much. Have a nice day.